I'm so glad to be here today, and I'm so happy that you all turned up for this. I think that's a testament to people are curious and excited to learn about science in America. And so that's what this is about today. But I'm actually going to start with another family picture. This is us in Olympic National Park. What a beautiful state you all live in. I'm very jealous. It's gorgeous up here. This is one we visited a few years ago. And I'm showing it because my story in science is not just professional, it's also personal. Science is personal for all of us. And I think many in this room know this story. But this is my sister Kristen, who, as my mom mentioned, is just amazing. She's brilliant and funny, and she's so cool. She works at NASA, you know, she's got it all. But when she was 34, she was diagnosed with breast cancer. It was a huge shock to all of us because there's actually not a history of breast cancer in our family, and she was only 34. And so you can see her hair is still short here from treatment. And I'm happy to say that she's okay now. She's cancer-free. And you know, scientific research saved her life. And it saved her life over her lifetime. The research that occurred during her life saved her life. And so, you know, breast cancer is very common. The incidence is 12%. And so I think, has anyone known someone affected or been affected themselves? Yeah, many people in the room. So it's very common. But I don't know if you know the good news, which is that since 1975, mortality is down by 58%. That's pretty amazing. Who was alive in 1975? I think all of you, right? Yeah. It's just a requirement of living here, I'm told. So I mean, in all of our lifetimes, we've had amazing progress, and I think sometimes this gets lost because everyone is talking, oh, why can't we find a cure for cancer? We have found 100 ways to treat what is in fact hundreds of different diseases, right? And cancer is not one thing, and we have made strides in all of these different maladies, and I think that sometimes we're so caught up in what we don't know now, we forget how far we've come. So this is a story that's both professional and personal, and I want to tell you how we got here. So science in America. I love doing science here, and I can say with confidence, this is the best place to do science in the world. I feel very strongly about that, and I think most of my colleagues would agree. There's nowhere like it. And so I want to tell you why. How did we get here? How does it work? How does the biomedical research infrastructure work? And then what does the future hold? So why do I think doing science in the United States is the best? Does anyone know how you lead the world in biomedical research? Anyone have an idea? Money. That's it. It's because we invest in it, and we have invested in it for decades. So let me show you a map of, this is a percent GDP spent on R&D, and you can see that there's just a few countries that spend more than 2% on R&D in the world. And of course, in terms of total dollars, the United States obviously contributes quite a bit. It really dwarfs almost every other country except for China. So we have historically put a lot of money into building this incredible research ecosystem in the United States. And these things take time to build, right? It takes time to get, this many people trained up and to do really amazing things, right? So doing amazing things, having big dreams, big ambition, that takes money and investment. And so I wanted to talk about how this started in the United States. And it starts with this man, Vannevar Bush, who has quite a legacy in helping build this in the United States. So he was an engineer at MIT, and he was appointed to lead the Carnegie Institute, and was head of the National Advisory Committee for Aeronautics. But in 1940, became head of the National Defense Research Committee. This actually didn't exist. He proposed it to Roosevelt, who apparently accepted his proposal immediately. Does anyone know why in 1939 this would be important to have a defense committee? Yes, war, right. So this is why when he went to Roosevelt, he said absolutely. Under his leadership, you might be familiar with one of the biggest projects he initiated. Did anyone see this movie? Yes, yeah, the Manhattan Project. It was one of the first big science projects, and it was because of his leadership that this happened. Now, we know that this project was successful, right? They achieved their ends. Of course, that end was a weapon. So however you feel about that, that has a strong moral consequence, I guess I should say. However, what they set out to do was ambitious, and they did it in a short amount of time. And so it worked. And so after the war, he continued to lead in scientific research and development and was chairman of the R&D board. And so what happened after the war is Roosevelt asked him, how can our scientific success in wartime be carried over into peacetime, right? And we don't want to do this just with bombs. Like, how can we actually take this model and apply it to other industries? And the answer is academic partnerships. So if you saw the movie, what you know was special is that the government put in investment and money and brought academics together to figure out this hard problem. And so it was Vannevar Bush who said, this is a successful model, and we should continue it in every aspect of this country's success. So his quote here is that scientific progress is one essential key to our security as a nation, our better health, to more jobs, to a higher standard of living, and to our cultural progress. So he was truly a believer in that investing in research was good for us, it's good for our country. So what did it get us? What has investing in basic research gotten us? It is the pacemaker of technological progress. So I don't know if anyone knows this, but it's the reason the internet exists, right? This is actually started with ARPANET. It was a bunch of researchers who wanted to share information across platforms, and it was done collaboratively. And so that was built in part with government resources. Now, of course, other people had a hand in making it, but I don't think the internet would exist in quite the same way if it had been, say, achieved through a private company, right? Would the World Wide Web exist if it were done privately rather than through this resource sharing, publicly funded mechanism? I don't know, but I think that obviously we all use the internet, and so we should be grateful that it exists. GPS, weather forecasting, so I don't know if you're aware, but the weather app on your phone gets its data from federally funded weather researchers, right? From NOAA, I think you've all heard of, right? So weather forecasting, and of course, healthcare. Healthcare is a big one, and that's actually going to be the focus of most of this talk, is where our basic research actually provides us with better healthcare. So just, did it work? Did investing work? Now, there's a lot of measures you could do this, but one easy measure is, Nobel Prizes. They're not perfect, but what they acknowledge is a key discovery. And what you can see in this graph is this dashed blue line. It starts going up really dramatically around about what time period? Do you see? It's small, but it's in the 41 to 60 range, right? So right after this investment was made is when you see this huge increase in the number of breakthrough discoveries occurring in the United States. And so again, there's a really strong correlation between how much we put into this and how much we get out of it, right? And so after Vannevar Bush started pushing this idea, it led to the formation of the National Science Foundation in 1950. Their goals are to promote the progress of science, advance the national health, prosperity, and welfare, and secure the national defense. It also led to the expansion of the NIH. Have you heard of the NIH? Yeah, everyone has. Okay, good. So the NIH started before this. It started with like a tiny little one-room lab, I think in the 1800s or something. But it was really, this is when sort of grants programs started expanding. And the goal of the NIH is actually to provide funding for research. And there's actually two sides of this. So the NIH is a physical place, shown here. They have a research campus in Bethesda. And this is what their intramural program within the walls. It is a massive, sprawling campus. They do incredible things there. There are 27 institutes. Each institute specializes in a different thing. So there's something, there's National Cancer Institute, there's the Neurological Diseases Institute, Heart, Lung, Blood Institute, right? So each of these systems has its own diseases, has its own special questions that need to be asked. And so this is how this works, is they get experts in all of these different things and have scientists researching all of these really diverse biological questions. And then within the NIH, what they really are good at is clinical research. So it is a facility like no other, where they're able to bring in people from all over the world to participate in clinical studies. So it's a really special place. And I'll tell you a little bit about some work done with collaborators there. But we should be so proud of this. This is, I really think, a crown jewel of our country. So what does the NIH do for you? Just to be straightforward about it, there were 356 drugs approved by the FDA from 2010 to 2019. So who wants to guess how many were directly funded by the NIH? How many drug development research? How many of those you think were required? All of them, pretty much, yeah. 354, so 99.4%. I would argue that those last two, it's possible there's probably some foundational research somewhere funded by the NIH. This is pretty remarkable, right? The NIH is the core funder of biomedical research in the United States. That's it. It dramatically overwhelms any other funding source. This is how all of us get our funds. And I will take questions, but we're going to do that at the end just to make sure we can, yeah. But if you have questions, keep them in mind, and we'll have a long time to answer questions at the end. Okay, so what does it take to get a new drug? I don't know if anyone has ever seen this pipeline here, but it takes a lot of work and a lot of money. So there's the first phases, which are just once you have a target. Let's say we know we want to target this, molecule or protein. Then you have to actually do the chemistry to find the molecule that will do this effectively and work in a human. You have to go through clinical testing to make sure it's safe and effective, and then it gets approved by the FDA. And this entire process can take 15 years, 20 years, right? And it takes millions to billions of dollars. So it is actually a lot of time and a lot of money to go from having a target to having a cure or a treatment, right? This is not trivial. But then you have to ask yourself, so this pipeline, of course, takes so many resources, but how do you even get to the beginning? How do you identify an appropriate target? And that is where basic research comes in. So I don't know if you've heard this term of basic research, but basic doesn't mean it's easy. What it means is that we're setting out to study the fundamental processes of how things work without a particular product or application in mind. Because in order to get to a target, you have to actually just understand how a system works, right? So fixing something that's broken is very difficult if you don't know what all the parts do, or you don't know how all of the molecules work together. So this is the area of basic research. And a lot of times in basic research, you're studying something that might feel esoteric, or it might feel really far removed from health or disease. And yet, this is where serendipitous findings come into play, and so often our best cures come from the most surprising places you can't actually predict. So it is critical that we have people studying everything, right? Because we don't know where the next cure will come from. And so what we do want is just knowledge about how the world works, and that will undoubtedly lead to better treatments, better healthcare, better cures. So I want to play this game with you. We are going to choose what project will you fund, okay? So I'll read them out to you. You can remember your color. So green is, how does algae move towards light? Pink is, what is in Gila monster saliva? How do bacteria stay safe from viruses? That's blue. I don't know if people know, but bacteria get infected with viruses too, but they're specialized. They're not the same viruses that affect us. And then red is, how do hormones affect chicken reproductive organs? Okay, so we're gonna go around the room, we're gonna vote. So who wants to fund the project studying how algae moves towards light? Anybody? Any takers? Okay, we have a couple takers. Who wants to know what is inside Gila monster spit? All right, we have a few, good. All right, who wants to know how bacteria stay safe from viruses? Oh, that's a popular one. Okay. Anyone want to know how hormones affect chicken reproductive organs? All right, we got a few takers. Okay, all right, now you get to find out what your money got you. All right, so if you wanted to understand how algae moves toward light, what you get out of it is a thing called channel rhodopsin. You don't have to remember that word, but it's this special protein they found that responds to light and creates a cellular signal. So what did this do for us? Well, just a couple years ago, Botondrowska actually put it in humans with retinitis pigmentosa, a disease of congenital blindness, and restored some of their sight. Incredible, right? It's the start. It's not complete, it's not finished, but it's the start of something very exciting. And then for me as a neuroscientist, this has been one of the most exciting discoveries in our field, because what it lets us do is actually control neurons with light. So for us, it's a tool to actually study the brain, Because if we want to know how specific brain cells work or specific circuits in the brain work, we can actually put this in them, flip on that circuit, and see what happens. That's incredible. So this has helped us make giant leaps forward in neuroscience to understand how the brain is working. Okay, who voted for Gila Monster Spit? All right, out of that you get Ozempic. Congratulations, you've made a ton of money. All right, these are the GLP-1 drugs. All right, this, I don't know if you know, but Novo Nordisk, the market cap on Novo Nordisk is bigger than the entire GDP of Denmark, where they exist. Wildly successful because of Ozempic, and Eli Lilly has been as well for related GLP-1 drugs. So these, as I think many people know from the news, are incredibly effective, just blockbuster drugs to help with diabetes, obesity, a range of metabolic disorders, and so much more. I don't know if you're following this, but it's pretty amazing. They think it helps with cardiovascular problems. There's actually thought now that it could help with neurodegenerative disease. I mean, it's wild. And so people are actually thinking that maybe it has broader effects on the immune system, but we're still really at the very beginning of figuring out how this works. So there is a lot more to come from these drugs. So it's pretty amazing. So why Gila Monster Spit, you might ask? It turns out, so Gila monsters are venomous, and I guess when they bit their prey, the blood glucose dropped or something that helped, kill their prey. And so someone thought, well, what's doing that? The GLP-1 was already discovered, but what happens with it is it gets broken down really fast. It doesn't last very long in the body. But in Gila monster saliva, there was this other molecule called exendin-4. It is the same thing, essentially, but it lasts a lot longer. So it was actually the first possibility that this could be a therapeutic came from discovering that molecule. And then, of course, since then, it's been built up to, now we have a lot of different, chemistry has been done to build new molecules. So pretty amazing. Thank goodness someone was curious, right? All right, CRISPR. Most of you voted for CRISPR. I hope maybe you've heard about this in the news. This is how bacteria defend against viruses. It's an ancient system that they use to remember a little snippet of a genome, so that if a viral genome comes in, they can snip it up, right? So what it lets us do now is target genes very specifically, and it is a revolutionary tool to edit the genome. So it will, and has already been, used to cure deadly diseases, genetic diseases. We can go in and actually change what's broken in a human. This is incredible. I mean, it is really, truly the frontier of genetic engineering, absolutely transformative. Okay, chicken reproductive organs. What you get out of this is hormone receptors. So these drugs targeting hormone receptors discovered in chick oviduct reduced breast cancer mortality by 71%. Now, you might know that, or might see that this is a different number than I told you before, and that's because it's targeting a specific type of breast cancer that's quite common, where these specific receptors are expressed. And so these are the drugs that saved my sister. It is so effective at treating this particular type of breast cancer that this is what's been responsible for this huge reduction in mortality. And so I'm very grateful for this discovery. And I think that it just goes to show you never know where your important discovery will come from. So it's important that we study everything. So now that moves me to my lab. So I started my lab 3 years ago, and I mentioned I'm a neuroscientist, but I really love physiology. I love how the body works. And a lot of neuroscientists kind of just live in the brain. They really only study the brain. I study how the brain actually senses the body. So again, I'm moving from basic research. I consider myself also mostly a basic scientist in that I'm not studying any one disease per se. I do study some diseases and pathologies, but what I really want to know is how things work. And so when you think of the senses, what you think of is typically the senses defined by Aristotle, right? How you smell, see, touch the world around you. So these are all senses coming from the external world, right? But this is not the full extent of your senses. You have a rich sensory world inside your body, and your brain is paying attention to all of this. So with every moment, your heart beats, your lungs expand with each breath. When you eat, your GI system is moving and churning and mixing. And of course, right now, your bladder is filling. You might be noticing it. A lot of the things that are happening in your body are actually mechanical movements, right? They're actually, the squeezing of your muscles or the stretching of different organs. And so this is what I focus on, is how do you sense mechanical force? And why is that important for physiology? So I study the GI tract and the urinary tract, both organ systems that move quite a bit. And I study them in both health and disease states. And I'm also interested in internal pain. So typically, you shouldn't really be feeling your organs. Let's hope that's the case, until maybe you need to go to the bathroom. But of course, a lot of people with diseases do feel their organs, right? People with IBD actually are in a lot of pain from normal GI movement. And so that means something has gone terribly wrong. And we don't understand a lot about internal pain and how that works. So figuring out, again, the mechanisms would be very, very helpful. And I also, the fun thing about running a lab is you get to decide on the directions yourself. And one of the ones I'm very excited about is actually using the tools that we've developed to understand how the body affects the mind, right? We all know that how we feel inside affects our daily lives, right? And our mood. People with panic disorder will actually start describing their panic attack by describing their body, right? Talking about their racing heart, the tightness in their chest, the churning of their stomach. These things are deeply tied to emotion. And actually we're at a point in neuroscience where we can study that in a very core way, like understand what it is, what signals are coming from the body to change our mood and emotion. So I'm also very excited about these directions in lab. And so, force sensing is critical for the function of multiple organ systems. I don't know if anyone has heard about proprioception. People call it the sixth sense. But this is actually how you know where your limbs are in space. So if you were to close your eyes right now and ask yourself, what is the position of your arm? You probably know if it's bent or straight, right? I don't know if you've thought about it before, but that's because you can actually sense how long your muscles are, how stretched they are, and that's what tells you how your body is positioned. This is something we take for granted, but of course it's how we move through the world fluidly. It's incredibly important for our daily lives. I mentioned heart beating, right? So you actually sense your own blood pressure. Hopefully, when you stand up today, you don't faint. But the reason you don't faint is because you have some stretch sensors in your carotid and your aorta, and as soon as you stand up, there's a drop in pressure. They detect this. They immediately tell your heart to speed up, and they tell your vasculature to squeeze so that it keeps blood in your head. Otherwise, you would faint. So hopefully that all works without you even thinking about it. And again, this is one of these amazing mechanical systems just working in your body all the time to keep you steady. Of course, I mentioned the bladder. The bladder fills and stretches. You are able to do this incredible thing, which is say, I should find a bathroom in like 10 minutes or 20 minutes. Like you can kind of gauge this, right? You don't think about it as an act of sense, and yet you know if you need to get to the bathroom, and you know if it needs to be now or if you have time, right? And that's because you can sort of sense how full it is. And similarly, I mentioned digestion. You feel full after a meal. All of these reflexes are occurring to actually move food through your system. So mechanical sensing is very important for the function of your body. So how do we sense force? Well, it's with this special molecule called piezo2. There's also piezo1. These are amazing force sensors, and the discovery of these is actually what won my mentor the Nobel Prize in 2021, along with David Julius. And together they discovered these special sensors for temperature and force that are in the body, and they mediate all these really important sensations, inflammatory pain, visceral pain, mechanical pain, but also these basic bodily functions like urination, respiration, blood pressure. I was fortunate in lab to be a part of the study showing that piezo 1 and 2 together mediate this blood pressure sensation that I mentioned, which was a long-known reflex, but no one knew how it worked. And I helped contribute to the story on mechanical pain. But my focus when I was in the lab was actually on understanding the contribution of these molecules to urination and that sense of bladder fullness. And really one of the, we use animal models to study the mechanisms for how this works. But one of the breakthroughs came through a collaboration. But so, when you hear about neuroscience, you don't typically think of the bladder, right? So I always feel like I have to tell people, well, why do we study urination at all? And the story I like to tell is actually begins with this man, Tycho Brahe. Has anyone heard of him? Okay, he's an astronomer. He was friends with Johann Kepler, apparently. So back in the 1500s. And he was, I think, around 1601, maybe. I might have the year wrong. He was at a banquet, and he didn't want to get up because he thought it was unbecoming to leave, even though he had to go to the bathroom. He just didn't want to get up at the banquet, so he held it the entire time. Got home later that night, and he had intense pain, and he could no longer urinate. And he died 11 days later. He actually had long enough to write his epitaph, which was, "He lived like a sage, died like a fool." And so I tell you this to tell you, if you need to go, I support you, right? So you need to listen to your body. It's important, okay? We don't want to lose anybody here. So urination is critical. We don't think about this. You cannot live without urinating, right? If you don't urinate, you die. Unfortunately, I hate to break the news, most people will have some lower urinary tract issue. And it's because so many things can go wrong. You can have overactive bladder, which is incredibly common. You can have incontinence, underactive bladder. Men have a lot of prostate issues. Anyone who gives birth also can have changes. So there's a lot of things that can happen to the urinary tract, and this really deeply affects your quality of life. You go to the bathroom multiple times per day, right? So if you feel like you always have to go, it is always something that you have to think about, and this is a real problem for people. We don't have great drugs to treat this. It's interesting to me because I study mechanical interoception, and as I mentioned, the bladder stretches and squeezes. Its whole purpose is to stretch, right? So it's a really great system for me to understand mechanical force sensing. And there's a lot of unknowns about how this really fundamental process happens. And so for me, it's a really rich area to explore how the brain senses the body. So, one of the first indications that we had that piezo2 might be involved, the special molecule, was when I talked to my dear friend and colleague at the NIH, Alex Schessler, and he said that they had individuals who actually had a mutation in piezo2. He had already characterized these individuals. Again, he's at the NIH, and he, along with Karsten Bonneman, a child neurologist, actually found out that there was this one individual who, she had issues and went to a really amazing doctor who decided to sequence her genome and found a mutation in this gene, and he didn't know much about it, but he went to Karsten, and Karsten figured out it was piezo2. And they did this incredible characterization of these individuals. And I want to say I'm really grateful to these individuals. This is an incredibly rare disease. They found several people around the world who had this very unique mutation, and they had really profound deficits. Those deficits helped us understand what this protein is doing in the body. So I like to point this out because it's a very rare disease. And you might think, well, why study rare disease? By studying a rare disease, we learned so much about the basic processes that work in the body. So for instance, these individuals came in, they have almost no sense of touch. So this is, we discovered this already using animal models, and it's confirmed that even in humans, piezo2 is the key sensor for touch. They also have no proprioception. So I'm going to show you this video where this woman was asked to do something that would be very simple for you and I. So remember that proprioception is that sense of where your limbs are in space. And she was asked to simply walk in a straight line with no visual cues. You can see she has a harness to help steady her. And she finds this very difficult. And this is, again, because she is blind to her own body. So the way she walks is she has to look to see where her legs are, right? And this is, again, something we take for granted. This sense of where our body is in space is incredibly important for us to move through the world fluidly. So by working with Alex's and Karsten's team, we found out that these individuals have no sense of touch, no proprioception, and remarkably, they never feel the need to go to the bathroom. So this, again, helped us understand that this molecule, piezo2, was really critical for that sense of bladder fullness. What was remaining is that they did have a sense of urgency. So if they felt they needed to go, it was an emergency. So that high threshold, that sort of in the pain domain, they could still sense. So that tells us a lot of things. It tells us piezo2 is important for low threshold stretch sensing, innocuous stretch. But there's something else that we still don't know that's detecting the high threshold stretch. So we are really, really grateful for this collaboration and to these individuals who contributed their time and effort to help us understand this. So this led us to a question. Given that stretch is so important for the function of the system, we wondered if you could actually change mechanosensation and improve bladder dysfunction. And so this is a story led by a really talented grad student, Yasmin Hamid, and she was begging me to do this aging study. And I was like, that is really hard. Studying aging is hard because you have to wait a long time, right? But I'm going to tell you a little bit about some exciting findings that are new in the lab. So as I mentioned, we use mice as a model system. They're incredibly important. But you might ask yourself, humans are special because we care about when and where we go to the bathroom. How could you study this in mice? All mammals care deeply about where they urinate. Have any of you had dogs? Yes. So your dog cares a lot about where it pees, right? Especially if it's a male dog. And so this is true of mice too. So I want to show you this video. I think it's very cute. She's sniffing around her cage, and then you see at one point, she has a thought. I think I have to go. And then she turns around, scoots her butt into the corner, and then she's done. Okay, looks, and she's all good. So it's kind of amazing. Like, they actually control where they go to the bathroom. They can sense when they need to go. They plan. They move to a corner, and then they go. So again, they have toileting behavior, is what it's called. So if you were to put a mouse, like shown here, just in a cage with filter paper on the bottom so that you can see where they went, what you see is that they'll urinate in the edges and corners of the cage, right? They're prey animals. They don't like to go in the middle as much, so they'll sort of stay in a safe area. And so we can see, Yasmin was like, I have these really old mice. 2 years old is pretty old for a mouse. So she's like, can I please just test them and see how their urination looks? So I said, go for it. And it looks like this. It's very different. You can see the pattern's different, right? There's a lot of small spots indicating some sort of leaking or incontinence. And then they don't control where they go to the bathroom as well. So you see they end up kind of having these leaks in the middle of the cage, or even large voids in the middle of the cage. That's really unusual. So it indicates they both can't control when they go, and they can't even necessarily control where they go as well. So we think it's actually a very useful model to understand urination. And so this is back to the idea that maybe we can use mechanical forces, or what we know about mechanical force sensing, to change the urinary pattern in aged mice. They have things that humans have: overactive bladder, enhanced urgency, even incontinence. So we wanted to rescue this by changing force sensing, and to do that, we used a special diet that changes the mechanics of the bladder. So this is before diet. This is an animal that you can see again has these little leaks in the middle of the cage. And this is just 4 weeks later, the same animal. And a lot of it's rescued. So it really improved their ability to control when and where they go to the bathroom. So we're very excited about this. Yasmin is still working on the mechanism of how this is acting in the bladder, but we're really eager to understand more because the current therapies that treat things like overactive bladder aren't well tolerated by everyone. So I have to acknowledge my lab. This is this incredibly talented group. They're from all over the world. I'm so lucky to work with them and mentor them. This looks silly. This is me asking them to do a series of silly tasks for our lab retreat. And they didn't know that I would take pictures and then put on a slide. Thanks for being good sports about it, lab. But I wanted to acknowledge a few people. Yasmin here in the corner, she did a lot of this important work in the urinary tract. We also are starting to track the movements of animals using machine learning to where we don't have to sit and score, and see, do they move this way? There's a lot of bias if you have to sit and watch an animal and score, are they in pain or not, right? So what Max and Scarlett over there on the side is doing is they actually built these, or implemented these incredible machine learning algorithms to track animals from videos and then watch, and then we can identify different behavioral sequences if they're in pain or not. This is, again, a tool for us that lets us study internal pain, which is incredibly difficult to study. The way people study pain right now is they might poke. That's not how you sense pain inside. It's spontaneous. So we're really excited about that. And I really wanted to also acknowledge my funding. The NIH has been key to my funding, and we'll talk about that more. And also the PrEP program, which funded Scarlett's time in lab, but it was cut mid-award. So I was told a couple of months ago that I'll have to supplement her salary now. And so we can talk a little bit about how this works and how we get money to run a lab, right? So how does this all function? I think this was the biggest surprise for my parents. It's a really opaque process. If you don't have someone who's in academia, it seems pretty weird because it's not like a normal position. So what happens is an institute provides startup funds. Often that institute is, wherever you go work, provides some pot of money. Often you also come with money. Maybe the institution that provides money is also the NIH because they have these transition awards. And so either way, you get a pot of money and they say, okay, with this money, you now have to produce knowledge. And we share that knowledge through publications. And so the cycle here is that hopefully with the new knowledge we get, we actually use those discoveries to then go back to other institutions like the NIH and ask for more money. So it's a cycle, right? And we say, okay, we discovered this, we've proven we can do this, and we think this opens up all these new questions. And so then you have to sustain your lab by getting continuous grant funding. So while, when you start a lab, which I did 3 years ago, you might have a pot of money to start with, that won't last you very long. And so the pot of money is supposed to give you enough data to then go to the NIH mostly and get more money to sustain. Okay, so really what people don't realize, it's actually not a straight line, right? So getting from money to discovery is it's a circuitous path. But really, it's kind of like running a startup company. Only we don't have a product other than knowledge, right? So the goal is to produce new knowledge about the world. So how do we do that? Well, that means we have to pay for everything, right? So we use this money to pay our own salaries. I don't know if you know this, but most scientists, they don't get paid more than maybe 30% from their own institution. It depends on the kind of institution. Some support more. If you're teaching undergraduates, for instance, you might get more support from your institution. But if you're at a med school, you get less. And so actually, we're expected to pay our own way through grant money. This funds expensive equipment like microscopes. I think people don't know how expensive it is to do science. My microscope is amazing. It costs $400,000, right? We use it all the time, and the pictures are beautiful, but it lets us see things that other microscopes can't see, right? It pays for the space, the facilities. It pays the salaries of my amazing scientists. It pays their fringe and benefits, right? Pays for reagents, for glassware. It pays for our animals and the care of our animals. It also pays for administration and facilities, like the building that we're in. It pays for, biomedical hazard waste removal, right? We have to comply with regulations. That's a good thing. We don't want scientists dumping chemicals everywhere. Right? We need help with this. And I point this out especially because this has been in the news a little bit, like how much administrative help do we need? And I want to point out that this is a very efficient operation. We run pretty lean, actually, and we have to pay for all of this stuff. And the way that works is that we have institutions that gather us all together, right? So we actually share equipment. There are core facilities, right? That instead of every lab having to buy a really expensive microscope, the university buys a really expensive microscope and says, you can all use this, right? Those core facilities are also funded by the NIH sometimes. The NIH has equipment grants, right? Where you can say, we want to buy this $800,000 equipment. No one lab could do that, right? So it's very efficient. All the facilities are combined. All the administration is shared, right? So it's actually a really wonderful way to make research happen, not in piecemeal, but all together. And also what that does is it facilitates collaboration. I have amazing colleagues. It's just like borrowing sugar from your neighbor, right? Only it's like literally like glucose from a Sigma or whatever, right? Like we actually do need to actually go next door and say, do you have this virus or do you have this, this reagent? So it's really helpful to actually be in a system where we're all gathered together and those facility administration costs are all shared. So I want you to know that for every dollar the NIH funds, this puts $2.56 back into the economy. So this is a really good deal for the US government, right? Because again, this creates jobs, it creates knowledge. As I mentioned before, the knowledge created is often built into companies. Those companies are bought by big pharmaceutical companies and end up leading to drugs, right? So this is a great investment, right? This is a good return on investment. So I also want to point out the opposite. If you take, or quote unquote, save a dollar, you lose $2.56 of economic activity, right? So the money you don't put in actually costs you money, right, in the long run. And so I want to point that out, right? Because this is, again, an amazing investment that our country now has made for many, many, many years and seen just really incredible paybacks for it. If you are interested in learning more, there's this great website, unitedformedicalresearch.org, and you can actually go state by state and see in your state, Washington's a big one, how many NIH dollars do you get? How many jobs does that create? And it's a fun little tool, so I encourage you to go look it up. Okay, NIH funding has been great. I want to point out, we have had broad bipartisan support for research funding in Congress. You can see that by this green line. It goes up and up and up and up every year, which is great. But it doesn't quite match inflation. So actually, when accounting for inflation, it's pretty flat. I like to mention, the main grants that sustain labs are called R01s, and there's a modular budget, which is kind of like the baseline budget. That value has not changed in 20 years. So the grant value, or the grant amount that someone 20 years ago was applying for is actually the same as what we apply for today. So we're expected to do a lot more with less money, but I'm a big advocate, actually spending more money pays back in big time. So while Congress has, like I said, broadly supported research support, I think we could do better. I think we could actually use a little bit more. And I'm going to tell you sort of what the process is to get this money. So grants equal innovation, right? That's clear. So these are awarded to scientist teams, and they're awarded for very carefully planned projects. And the reason they're awarded is it's gone through a competitive process of peer review. And so what this is, it starts with an idea, or maybe there's a specific funding call. And then after you see this funding call or have the right idea for the right mechanism, you write a grant, you submit a grant. Does anyone want to guess how long my R01 was that I just submitted? Like how many pages?
A lot.
It was a lot. It was 100 pages long, right? So this is not just I'm like, oh, hey, will you give me some money for this? Like, this is a well thought out project. We have a timeline, and R01s, for instance, which again, these are sort of the bread and butter grants that everyone hopes to get. They're 5 years. So we plan in sort of 5-year chunks of what we can accomplish. And so you have to look ahead and think what you will need, and plan out, who are you going to hire with this money? What will you spend it on? So it's a lot of paperwork, and there's a lot of different files. The one I just submitted was a collaborative grant, so of course we had sharing agreements, and we had a collaboration agreement, and there was a lot that went into planning this grant proposal. So one application, it's a lot of work to put these together. So you submit your grant, and then it is reviewed by a panel of experts. This happens months after you submit your grant. It actually takes so much coordination, and I want to point out this is all coordinated by the amazing people at NIH. Again, this is their extramural funding program. So instead of being the research they fund within their campus, it's the grant mechanisms that they give outside. Most of their money is spent on these external grants to universities. And again, the grant money is for scientist teams. So they gather experts from around the country that don't necessarily work at NIH. We all put in this service to review grants. They gather the right experts to then review all the grants, and NIH coordinates when this happens. They coordinate the meetings, and they have, of course, their own experts in different institutes that know sort of who to gather. They know what the funding priorities are, what the kind of ins and outs of that field are, it's complicated. So this is a lot of work to get this all together, and these review cycles happen multiple times per year for every disease and mechanism you can imagine. So they then assign a percentile score and an impact factor, and then they actually review and score, and then they recommend those with the top scores to a council, an administrative council, that then weighs kind of the scores that they have with the money available, and they set pay lines, right? So they say anything with this percentile and above or below gets funded, right? So smaller numbers are better, right? Does anyone want to guess the percent of grants that get funded are?
About 12%.
12? 20? Yeah. So it used to be more like 12. These days, the pay lines this year were 9% at National Cancer Institute and 8% at the Neurological Diseases Institute. So 8 and 9% means that if you're lucky, 1 out of 10 applications get funded. So you go through, this process, by the way, takes about 8 months. So you go through a lot, and the chance that you get funded is pretty low, right? So this is why I'm like, please, Congress, please give us more money. Because you can imagine, people put a lot of time, these are brilliant scientists, like there's a lot of good science being proposed that doesn't get funded. You're allowed to submit twice, and then you can't resubmit, you have to kind of start over. So it's a lot of work that you have to put in to get enough money to sustain your lab, right? This is why we spend a lot of our time writing grants. So I want to say federal funding has been critical to my research and training. I received what's called an F32. This is an award that helped my postdoctoral training, where it funded me for a couple years in my post-PhD research. These days, in my field and most biological research fields, you don't get a PhD and then get a lab. That's very, very rare / almost unheard of. There's like a few instances where that can happen, but usually you do a postdoctoral fellowship, which in the field of neuroscience lasts 5-6 years after your PhD, so it's a pretty slow process. We don't also get paid that much. Like, scientists put in a lot. We do this because we really love it and believe in the endeavor. And so luckily there are fellowships to help our training and help pay for our salary during training. I also got this wonderful transition award. They have these very cool mechanisms where they say, okay, you've proven yourself in your postdoctoral work, but you need a little more training, maybe 2 years to finish up, and then we'll also fund you to start your own lab. This is before we have a position. That is a really special thing, because then when you're on the job market, you can say, you know, the NIH says that this is valuable, that my research program that I've planned is valuable. And so it actually funded the first 3 years of my lab. We just had our 3rd birthday, so that funding is just coming to an end. But I was really grateful for it. And you know, at the beginning, it's hard to get funding, because as I mentioned, to get funding, you need data. But getting data takes time, right? So this early phase of your career is actually pretty delicate. You have some startup money, but this is when it's, you know, hard to get going and you need money to sustain yourself. And then ultimately the goal for almost every scientist is to get an R01. This is really kind of like I said, the bread and butter funding for a lab. This is the one that the modular budget hasn't changed. It is a 5-year grant. The modular budget, I think, still sits at $250,000 a year. That sounds like a lot of money, but remember, it has to pay my salary, the salary of anyone who works on those projects, which is usually multiple people, and fringe, which is 30% over their salary, and it has to pay for reagents and animal costs and all of that, right? so one R1 actually at my med school is not enough. Can't make it, I think. With one's really pushing it. You'll have a very small lab. So the good news, the very exciting news, is that my first R1 score came back. This was a grant I put in. I know it's very tiny, but I'll tell you the numbers. The grant that I put in with my colleague, the first time we put it in, it was triage, which means it didn't get reviewed. And the second time we put it in, I was so excited. We got a 2 percentile. So that's below the pay line. We were like so excited. And I'm not bragging. This is really just like, this was one of my best days. It was like my colleagues believe in what we're doing. Right? My peers reviewed this and said, yes, you two together, you can do this very ambitious project. I was so excited. This score was received in October. That's when they reviewed it. We submitted it last summer and then got the score in October. And then there was a council meeting date that's set on the books for January, but that didn't happen because all the NIH meetings got canceled. And it still hasn't happened to my knowledge. So everything has been delayed and pushed back. So if council doesn't meet, they can't give you the money, so we don't have this money. I'm still hoping it comes through. But my colleague is at Harvard, so they might just pull it. We don't know. But of course, I hired people back in October, knowing that this money was going to come, in maybe March or April, and it hasn't. So it's a little tricky, right? You have to sort of plan ahead, because training people takes time. So you got to find the right person, hire them, and get them trained. So I did the hiring before I had the money, because that's actually what you're supposed to do. So yeah, we'll see what happens. I don't know. But the funding to sustain my lab is a little bit in limbo right now. But I hope that it comes through. So what is the future of science in America? We don't know. It's all a little uncertain right now. There's a lot of upheaval, a lot of changes. But I'll tell you just the plans that have, these are not congressional approved plans. These are just the plans that have been leaked or proposed by the White House. And it is NIH budget cut by 44%. 1,200 NIH employees have been fired, and there's really uncertain future for others. As I told you, my dear colleagues are at NIH. One has already decided to leave because half his team was laid off against his will. Active grants have been terminated. Hundreds have been pulled. I told you one of my students' grants has been pulled. New grants are delayed or not awarded yet. The proposed NSF budget cut is by 55% with the proposed 50% layoff. So you can see how that might affect everyone in science. So I guess I urge you to think about how will this affect you. I told you a story about my lab, and so it's pretty clear how that might affect my lab. If the pay lines are already at 8 or 9% and you cut 44% of the budget, it gets a lot harder. But it's also personal, right? Science saved my sister's life, and that didn't happen with research that occurred a few years before she had her disease. That research occurred in the 70s, 80s, 90s that led to saving her life later. And so what research gets lost now, it won't affect you next week, it won't affect you next year. It'll affect you in 10 years from now, 20 years from now. These things are slow and they take time, right? And so we don't know what we'll lose if we cut research heavily. But we will be less healthy and we'll be less well off for doing so. So I am just so proud to be doing research here. I think we should all be so proud to be in the United States where these incredible things are happening. I am truly excited about the precipice. Like, we are on the precipice of so many important discoveries. We have all these amazing tools I told you about. CRISPR was not discovered that long ago. I mean, there is a lot of cool stuff on the horizon. Ozempic, this hormone was known about for a long time. These drugs are kind of newer, right? And so we are really at the start of exciting things, and I'm just so proud to be a part of it, and I just love my job. And so thank you so much for coming.